Hi everyone, my name is Julia and I'm down here at the Coastal Biology campus in the greenhouses with Erin Aiello to interview her a little bit about her research. So Erin, would you just give a rough introduction of yourself and what sort of science you do?
Sure. I'm Erin. I am a third-year PhD student in Ingrid Parker's lab, and I think that my research most concisely could be talked about as community ecology. So I study the interactions between plants and soil microbes, and that includes the direct interactions between plants and soil, but also the indirect interactions between plants via their soil microbes. So,
Very, very cool. I think you were about to answer my next question, which was to expand a little bit on what your specific research project is and the types of interactions you study.
Thank you. So when I'm talking about these interactions, there are many interactions in the soil between soil biota and the plant, but I'm going to talk primarily about mycorrhizal and bacterial interactions. And so mycorrhizae, or mycorrhizal fungi, are fungi that can either wrap around the root or penetrate the root and the cells in the root of the plant and exchange nutrients for photosynthate. And so the plant will photosynthesize, create these sugars, and those will be flowing through the plant to feed the plant cells. And the fungi will take advantage of this and consume these sugars. And the fungus will break down phosphorus in the soil, mostly, many other nutrients, but phosphorus is the most important to plants of those nutrients. And it will, by diffusion, absorb these nutrients and give them to the plant in exchange for the photosynthate. So the bacteria, these are mainly Rhizobia and Bradyrhizobia species, and they break down nitrogen, they fix nitrogen in the soil into forms that, that the plant can use. And so typically nitrogen is found in forms in the soil that plants can't use. And they turn it into a different form, the plant benefits from that. And again, in exchange for photosynthate. So the plant actually has special structures on its roots called nodules. They're like a little house for the bacteria. So they protect the bacteria, and also it really facilitates that relationship. Okay. So the reason that this influences invasion and competition is because sometimes, well, not only do they benefit and make the plant a better competitor, but they can also be shared. And so in my research, I'm looking at how the shared mutualists and potentially shared pathogens influence the competition and the ability of the invasive plant to establish. So the overall effect, whether it's facilitation or inhibition of the invasive plant or the native plant, really depends on the balance of these pathogens and mutualists in the soil. So which one is having a stronger influence on the other.
And do you think these interactions are going to be positive for the invasive or maybe negative for the native? Or
Yeah, so if they share mutualists and the native plant is able to, well, this is very muddy, right? So I don't know the answer, which is why I'm studying it. But if the native plant is able to harbor these mutualists that the invasive plant requires, then that would be an indirect positive interaction, indirect positive influence on the invasive plant, and indirectly negative for the native plant since it helps the invasive plant. And then if it's pathogens they share, then this would be an indirect positive influence on itself. If the native plant were to be able to harbor these pathogens that the invasive plant is not adapted to resist. And so that would negatively influence that plant indirectly and then circle back and positively influence the native plant. And then the opposite can also occur. So the invasive plant can take its pathogens with it, and those pathogens the natives have never seen before, and so they're not going to be adapted to fight those off, and that will indirectly negatively affect the native plant, and indirectly, on another level, positively affect the invader.
Really interesting. Lots of interactions there. It makes me wonder about facilitation, and I wonder what sort of mechanism of facilitation you say or you might think would be associated with your study organisms, with legumes.
So I'm not going to talk about the broom plants here because it's so muddy when they're invasive. But typically, legumes are thought of as a facilitative species. Now, in succession, the three models of succession, facilitative, inhibitive, or tolerating, tolerance. So it's never that cut and dry. It's usually a combination of all things happening all at the same time. And so a legume can increase the nitrogen in the soil because of its relationship with bacteria. So that can be facilitative of other species in the area. Can also be facilitative of invasive species. So that adds another level. But they can also be really strong competitors because of this ability to grab a lot of nitrogen and grow really quickly. And so, yeah, it's muddy. It's also, this is ecology. It's not physics.
Sounds like there's lots to explore.
Yeah.
Well, very cool. Thank you so much for sharing about your really interesting research.
Thank you, Julia. And thanks everybody for listening.